Hey there, everyone. We are about to start a new chapter in our study of biology, and this chapter is coming on the heels of our discussions of the cell cycle, mitosis, and meiosis. And of course, one of the things that we really understood about those processes is the importance of faithfully replicating the DNA and just working with the DNA in general. So this chapter is going to give us a lot of insight into some things that we probably already know a little bit about, but things we definitely need to know more about: the structure of DNA, the function of DNA, and then we'll delve into a little bit of DNA replication, which we obviously talked about during S phase of both mitosis and meiosis, and then we'll finish up by talking about DNA repair mechanisms and the nature of mutations to the genome. Okay, so let's start by reviewing what we know about DNA so far. So we know that genetic information in all cells is going to be contained within DNA, which of course is a nucleic acid that is built by attaching a certain type of monomer together by dehydration synthesis. So do you remember what kind of monomer that was? Well, if you say nucleotide, you are correct. So nucleotides are attached together by dehydration synthesis, and doing this over and over and over again will eventually give you a nucleic acid, whether that is DNA or RNA. If you don't feel real confident in the concept of monomers being attached together to form polymers, then you might want to go back and review Chapter 3. Okay, so we also know that DNA maintains a double helical shape as opposed to RNA, which is single-stranded, and DNA is held together by a certain type of chemical bond between bases that allows the two strands to be held together. So if you don't remember, this type of chemical bond was, of course, hydrogen bonding. So DNA contains 4 different types of nitrogenous bases that are a part of the different nucleotides, and these bases are adenine, thymine, cytosine, and guanine. RNA contains the same bases, except for thymine, which is replaced by uracil. We also know that cells maintain their DNA as condensed chromosomes that are stored within the nucleus if you're talking about eukaryotes, or within a singular nucleoid space if you're talking about prokaryotes. And we also talked in the previous chapters that eukaryotes have multiple linear chromosomes, whereas prokaryotes tend to have just one singular circular chromosome. And we know, of course, that chromosomes have to be duplicated in order for cell division to occur. And finally, we know that chromosomes contain genes, which are discrete sequences of DNA that provide the information necessary for the cell to build all of its proteins. Okay, so before we actually start looking at the specific structure and function of DNA, I want to go through a little bit of history of how we came to our modern understanding of what DNA is and what DNA does. I think it's always important to not take the information that we have currently for granted. We want to have a good understanding of how we actually came to know what we actually do know. Okay, so DNA was discovered back in the 1860s by a physician named Friedrich Miescher. So basically what he did is he took a blood culture and he isolated the white blood cells, and then he was able to lyse these cells. And what he did is when he took a nuclear fraction, he was able to isolate phosphate-rich compounds out of these white blood cell nuclei. Now, knowing what we know currently about the structure of DNA, especially the fact that every nucleotide contains a phosphate, we can probably surmise that yes, it was DNA that he isolated out of these nuclei. Not only because we know DNA contains phosphates, but because DNA is the main thing that you find inside the cell nucleus. Okay, but that didn't really tell us a whole lot other than that was kind of our first recorded instance of knowing that we're probably dealing with DNA. So if you fast forward about 60 or 70 years, a man named Frederick Griffith performed a number of experiments that actually demonstrated that some kind of hereditary molecule could be passed from cell to cell. Now, we're going to see that he didn't necessarily know that this was DNA, but knowing what we know, we can, of course, infer that, yes, it is DNA that he's talking about. So let's talk about those experiments. He had 2 different strains of a type of bacterium. He had a non-pathogenic R strain, which even if you get exposed to it, you're not really going to get sick and die. And then a pathogenic S strain, which if you do get exposed to it, you are going to get very, very, very sick. So the idea here is that the S strain is different from the R strain because the S strain synthesizes a capsule that coats the outside of the cell wall and allows this bacterium to escape the immune system, meaning that if you do get infected with it, your own immune system can't really protect you from it, and you are going to get very, very sick. Whereas the R strain, since it does not have this capsule, it is very, very susceptible to immune system surveillance, so your immune system should be able to take care of it in the case where you actually do get infected. So here is the experiment that he did, since he had both of these strains available to him. He took several different groups of mice, and he injected them with different strains of this bacterium. In one case, he heat-killed the S strain, so the very pathogenic strain. He exposed to very high levels of heat, which killed the bacteria, meaning that all that was basically left over were the dead husks of the bacteria and anything that was inside of those bacterial cells. And then he also had a living R strain that was not heat-killed. So he injected one group of mice with the heat-killed S strain and another group of mice with the living R strain. And what he found is that when he co-injected these so the idea here is that if he just injected the heat-killed S strain, the mice were not going to be affected because the bacteria are dead. They can't really do anything at that point. If he injected just the living R strain, the mice will be fine for reasons that we just said. But if he injected both of these bacteria together in the same group of mice, the mice were killed. So the conclusion that he came to is that, okay, there must be something about the dead S strain bacteria that is being passed on to the living R strain bacteria. And we know the living R strain bacteria just on their own are not going to kill the mice. The killed S strain bacteria just on their own are not going to kill the mice, but there's some synergy of injecting them together that actually causes the mice to die. So what he did is he isolated the living bacteria from these mice and basically found that, okay, these bacteria took up some kind of hereditary molecule from the dead S strain cells, and those got passed on to the R strain cells, and that transformed them into living S strain cells. So, to repeat myself on that because I may not have been totally clear, the dead S strain cells have their DNA molecules that provide the instructions on how to make that capsule that evades the immune system. They were able to pass on their DNA to the living R strain bacteria, and in doing so, that facilitated a process called transformation. Transformation is when a cell takes up a piece of DNA that it normally does not have, and it begins expressing its genes and takes on new physical characteristics. So those R strain cells that normally would be susceptible to the immune system were transformed into S strain cells that then went on to kill the mice. Okay, so at that point, that was a very, very important watershed moment for us, but that did not definitively prove that it was DNA that was the hereditary molecule. At this point, there were two candidates. Either the hereditary molecule was DNA, or it was a protein. So, the next important set of experiments to distinguish between these two possibilities was led by a group of scientists led by Oswald Avery. And what he did is he isolated nucleic acids, so both DNA and RNA, and he isolated proteins from that same S strain bacterium. So on the one hand, we have isolated nucleic acid, and on the other hand, we have isolated protein. So he took these two fractions and treated them separately with enzymes that will degrade them and break them down. So nuclease enzymes will chop up and degrade the DNA and RNA. Protease enzymes will chop up and degrade the protein. So when he chopped up and degraded the nucleic acid, he found that transformation was no longer possible, meaning that if he exposed R-strain cells to degraded nucleic acid, they could not be transformed and they could not go on to kill any mice. But if he did the same thing, where the only thing he's chopping up and degrading is protein, this had no effect on transformation, and the R strain was still transformed into S strain. Meaning, the conclusion here is that the only way that transformation is possible is if that nucleic acid is still intact. It doesn't matter if the protein is still intact, it matters if the nucleic acid is still intact. So the clear conclusion from this is that the hereditary molecule that we saw on from a Griffith's experiment, it has to be some kind of nucleic acid, either DNA or RNA. But a lot of people still did not believe this because they saw DNA as being too simple of a molecule to carry such complex information. You have to remember, DNA only contains 4 different types of bases. So think of it this way. There's 26 letters in the English alphabet, right? So you can make hundreds and hundreds and thousands of different types of words by putting those 26 letters together in a variety of different combinations. Well, how much information could you really get out of using an alphabet that only has 4 letters in it, like DNA? So DNA has A, T, G, and C. So how much information can we really make out of such a simple alphabet? So a lot of people still, in spite of Avery's experiments, they still saw proteins as being more likely to be the carriers of this complex information because there were 20 different types of amino acids that make up a protein, which is obviously a lot closer to a 26-letter alphabet. So this quandary is actually something that we are going to clear up in a later chapter when we start talking about the genetic code. But basically, what you should take from this is that, yes, it is true that DNA is the genetic hereditary molecule, and there is a way for it to carry complex information in spite of the fact it only has 4 bases. So that is something that we will address in a later video. Okay, so let's definitively prove here that DNA is the hereditary molecule and not protein. So this is the Hershey and Chase experiment. So to understand how this experiment was performed, I'm going to ask you a couple of questions. The first question, what elements are found in a DNA molecule? So if you need to pause the video here, go ahead and do so. Go ahead and go back to chapters 2 and 3 and answer this question before you move on. So if your answer were carbon, hydrogen, oxygen, nitrogen, and phosphorus, then you are correct. Those are the 5 different elements that you'll find in every DNA molecule. Okay, so next question, what elements are found in a protein? You may want to pause the video and go look that up again. So for a protein, 4 out of the 5 elements are the same, carbon, hydrogen, oxygen, and nitrogen, but proteins do not contain phosphorus. Instead, they contain sulfur. So basically, Hershey and Chase took advantage of this fact and designed an experiment in which they used radioactive isotopes of both phosphorus and sulfur, P32 and S35, and that's going to be the basis for how they distinguish between DNA and protein. Any molecules that incorporate radioactive phosphorus must be nucleic acid, and any molecules that incorporate radioactive sulfur must be proteins. So if you look over here, what they did is they took a type of virus called a bacteriophage. This is a virus that exclusively infects bacteria. So these viruses have 2 major components to them. They have an outer protein coat, so that's just made of protein, and then they have a DNA core. So you can see the coat here in red and the DNA here in green. So they were able to grow these viruses either in the presence of P32 or in the presence of S35. So in doing so, they either made the protein coat radioactive or they made the internal DNA radioactive. So they took these 2 different fractions of virus, one in which the protein is labeled, another in which the DNA is labeled, and they allowed these viruses to separately infect bacteria. So after the infection occurred, they did a couple of centrifugation steps so that they're only isolating the bacteria and not the viruses. They found that the viruses that were infected with the P32 labeled phage viruses, these bacteria were radioactive. Whereas the bacteria that were infected with the S35 labeled viruses, they were not radioactive. The idea here is that the hereditary molecule that these viruses are injecting into the bacteria to allow for the production of new viruses, it has to be DNA. It cannot be protein. So DNA, when a virus infects a cell, DNA is passed on to the infected cell, but the protein is not. You can see here the S35 labeled protein capsule stays on the outside of the cell, but the P32 labeled DNA is injected into the cell. So this was a very, very important experiment, especially for the doubters and the skeptics that we mentioned before. This definitively proved the hereditary molecule cannot be protein. It has to be DNA. Alright, so that is going to do it for this video looking at kind of the historical basis of our modern understanding of DNA and how it works. So join us next time and we are going to look a little bit more of the history of how the double helix was discovered by Watson and Crick and company, but we'll also delve a little bit more into the actual structure of DNA, and then in later videos we'll start looking at DNA replication and DNA repair. Thanks again, and I will see you next time.